Hello everyone. Today we are going to start learning about immunology. So I'm going to use notes from EPG Padashala, which is a government site of Government of India. So you could see the notes, and I would like to share my sincere gratitude to people who developed this note. You could see their names and position. Thank you. So this topic is about overview of immune system, components of the immune system, principles of innate and adaptive immunity, some basic ideas. So just start. So today we're going to learn about immunology, components of the immune system, principles of innate and adaptive immunity, innate immune response, adaptive immune response, active immunity, passive immunity, and inflammation. And here we have some sort of learning objectives like historical perspectives of immunology. We'll be learning early theories of immune response, selective theory, instructional theory, clonal selection theory, then innate or natural and adaptive or acquired immunity, adaptive immunity or acquired immunity or specific immunity in detail, then immunity active, passive, and delayed type, along with a quick summary of all these topics. So let's start. How the branch of immunology has originated, basic concepts of immune response, what is specific and non-specific immunity, understanding of active and passive immune response. So first let's have some perspective or historical perspectives of immunology. The word immunity, immune is free of or to accept, was used in the context of being free of the burden of taxes or military conscription. The history of immunology is really slightly more than 100 years of if one considers Louis Pasteur as the father of immunology. From early writing, it is clear that primitive man knew about disease and its ravages. The records of the presence of pestilence and disease can be found in Babylonian epic of Babylonian epic of Gilgamesh. Of other more recent writings from old dynasties of ancient Egypt, one finds even more descriptions of disease. From these writings, it is apparent that man knew that once he had survived the disease, he will normally not able to get that disease again. The science of immunology grew from this knowledge that those who survived infectious diseases rarely contracted that disease again. This was an observation that was made long before the establishment of the germ theory of disease. In 430 BC, Caesarius, I'm not sure about the pronunciation, recorded that while the plague was raging in Athens, the case of sick was being taken by the individual who had already contracted the disease and recovered. Beginning around 1000 AD, the ancient Chinese practiced a form of immunization in which they inhaled dried powders derived from the crusts of smallpox lesions. Around the 15th century, a practice of applying powdered smallpox crusts and inserting into the skin became common practice. This process was referred as variolation and became quite common in the Middle East. Variolation, don't forget that. Lady Mary Wortley Montagu in 1689 to 1762. Lady Mary was the wife of the British ambassador to Turkey. She was a smallpox victim. She variolated both her son and daughter in 1717 and 1721 respectively. Initially, there was much resistance and criticism by the higher society in England, but later on, the permission to vaccinate the children of the Prince and Princess of Wales in 1772 dramatically promoted the adaptation of this method in England and in other part of Europe. Ultimately, the process was popularized in Great Britain. It was strongly opposed by church and was highly downcast, particularly if one were Christian. Next, we are going to learn about Edward Jenner. By the second half of the 18th century, it was noticed that women who milked cows were safe from smallpox disease. Numerous undocumented accounts show that the connection was made between contact with cowpox virus and protection from smallpox. Taking clue from this, Edward Jenner started thinking about vaccine. After years of experimenting, in 1796, Edward Jenner carried out a famous experiment. He deliberately exposed the boy who has recovered from the chickenpox to virulent variola virus 2 months later and found that the child was protected, showing only a mild inflammation around the site where the virus was injected. Jenner wrote a paper in 1798 explaining his experiments and wanted to report his first case study in the Transactions of the Royal Society of London. His study was rejected. He then went to London to demonstrate his theory. No one would submit to his vaccination. Discouraged, Jenner returned to Berkeley. In 1801, Jenner published The Origin of the Vaccine Inoculation, describing how cowpox virus was prepared and used to protect or vaccinate healthy persons against smallpox. Vaccination, the word Jenner invented for his treatment from the Latin vacca, means a cow, was adapted by Louis Pasteur for immunization against any disease. Louis Pasteur in 1822 to 1895. Louis Pasteur developed the germ theory of disease. Pasteur's work led him to the development of pasteurization or heating as a means of stopping fermentation. He was the first one to grow in vitro the culture, the infective organism of fowl cholera. In his experiment, he inoculated the culture of bacteria to chicken. Some of them survived and many of them died due to infection. He then went for the vaccination. On coming back, he injected the leftover culture to the chicken who had survived from the previous infection. To his surprise, no one got infected. He reasoned that it could be because the culture which he had used for the second time was lost its virulence because of aging. There is development of resistance to infection in the chickens because they had survived the first infection. So, this was the start of using attenuated microbes for providing immunity against infection. He also found that rabies was transmitted by agents so small they could not be seen under a microscope, thus revealing the world of viruses. As a result, he developed techniques to vaccinate doses against rabies and to treat humans bitten by rabid dogs. Robert Koch in 1843 to 1910, a German physician who worked on anthrax 1870s and found out the causative agent of an infectious disease. In 1882, Koch was able to demonstrate that the germ theory of diseases applied to human ailments as well as animals when he isolated the microbe that caused tuberculosis. So, Koch's pustules are still used to identify infectious organisms. Postulates. He was awarded 1905 Nobel Prize. Emil Adolf von Behring in 1854 to 1970. Emil A.V. Behring, a student of Koch, along with Kitasato and Wernicke, discovered antitoxin for diphtheria and tetanus and applied it as therapy. So, Emil von Berling and Shibasa Shibasaburo Kitasato were able to demonstrate passive immunity when they took serum from animals infected with diphtheria and injected into healthy animals. These same animals were found to be resistant to the disease. Ultimately, these serum factors were acknowledged in 1930 as antibodies. In 1901, von Behring was awarded first Nobel Prize in Physiology. Then Paul Ehrlich in 1854 to 1950. Ehrlich hypothesized that antibodies are specialized molecular structures with specific receptor sites that fit each pathogen like a lock and key. Thus, the first insight that the body has a specific defense system was introduced. He also realized that this powerful effector mechanism, which is used in host defense, could cause severe tissue damage if turned against the host, and he termed this as antitoxicus. He shared 1908 Nobel Prize with Mechnikov. I'm not sure about the pronunciation of the scientist. Sorry for that. Then Eli Mechnikov, 1845 to 1916. He is an embryologist and studied the starfish development. He was the first one to found Phagocytosis and birth of cellular immunology. Phagocytosis is the process of eating any foreign body. He shared Nobel Prize with Ehrlich in 1908. So he shared Nobel Prize. Then Sir Frank Macfarlane Burnet. He described a clonal selection theory to explain tolerance. He received 1960 Nobel Prize for the discovery of acquired immunological tolerance. Rejection of donor grafts was due to an immunological reaction. The clarification of the immune response and the science of immunology did not progress in a systemic or chronological order. Nonetheless, once scientists had a basic understanding of the cellular and humoral branches of the immune system, what remained was the identification of the various components of this intricate system and the mechanisms of their interaction. This could not have been accomplished without the concomitant development of molecular biology and genetics. Early theories of immune response. Below is a history of antigen antibody theories. Selective theory. Selective theory states that an antigen reacts with an already existing antibody. The earliest conception of selective theory dates back to Paul Ehrlich. In order to explain the origin of serum antibody, he proposed that cells in the blood express a variety of receptors, side chain receptors that could react with infectious agents and inactivate them. Ehrlich proposed that binding of the receptor to an infectious agent was like the fit between a lock and key, and he suggested that interaction between an infectious agent and a cell bound receptor would induce the cell to produce and release more receptors with the same specificity. The specificity of the receptor was determined before its exposure to antigen, and the antigen selected the appropriate receptor. All aspects of Ehrlich theory would be proven correct with the minor exception that the receptor exists as both a soluble antibody molecule and as a cell bound receptor. Then instructional theory. Instructional theory suggests that antigen. Wait a minute, I'm quite confused about the selective theory. Once again, I'm going to read that. In order to explain the origin of serum antibody, he proposed that cells in the blood express a variety of receptors, side chain receptors that could react with infectious agents and inactivate them. Ehrlich proposed that binding of the receptor to an infectious agent was like the fit between a lock and key, and he suggested that interactions between an infectious agent and a cell-bound receptor would induce a cell to produce and release more receptors with the same specificity. The specificity of the receptor was determined before its exposure to antigen, and the antigen selected the appropriate receptor. All aspects of Ehrlich theory would be proven correct with the minor exception that receptor exists as both soluble antibody molecule and as a cell-bound receptor. An instructional theory. Instructional theory suggests that antigens play a vital role in determining antibody specificity. According to the instructional theories, a particular antigen would serve as a template around which antibody would fold. The antibody molecule would thereby assume a conformation complementary to that of the antigen. Then, clonal selection theory. Clonal selection theory is the most accepted theory given by. Wait a minute, I'm quite confused about the instructional theory. I'm going to once again read that. Instructional theory suggests that antigens play a vital role in determining antibody specificity. According to the instructional theories, a particular antigen would serve as a template around which antibody would fold. The antibody molecule would thereby assume a conformation complementary to that of antigen. So there must be some sort of conformational change to antibody. Okay. That is something which is explained with the help of instructional theory. Then clonal selection theory. Clonal selection theory is the most accepted theory given by Niels Stern, David Talmage, and F. Macfarlane Burnet. According to this theory, an individual's lymphocyte, B or T lymphocyte, expresses membrane receptors that are specific for different antigen. The specificity is determined before the lymphocyte is exposed to the antigen. Binding of an antigen to its receptor, specific receptor activates that particular cell, leading to its proliferation into a clone of cells that express the same immunologic specificity receptor as the parent cell. So here we could see a picture which illustrates this thing of clonal selection theory. Now we mentioned important aspects related to the history of immunology. So now we are going to learn some other aspects like innate and natural adaptive or acquired immunity. So let's try it. An immune system is a system of biological sources and processes within an organism that protects organisms against diseases. And this is something which you need to carefully learn, because this is something which is happening inside of your body and inside your pet's body, or inside all sort of organism's body. But today we will be specifically learning about human immunology. Okay, so this is some medical aspects also related to immunology. So this will be a lecture series which I'll be reading. So come, let's explore the topic together. An immune system is a system of biological structures and processes within an organism that protects organisms against diseases. It detects a wide variety of diseases causing agents from virus to parasitic worms, and needs to differentiate them from the organism's own healthy cells and tissues in order to function appropriately. Simple unicellular organisms such as bacteria have enzyme systems that protect against viral infections. Other basic immune mechanisms evolved in ancient eukaryotes and remain in their modern descendants, such as plants and insects, including antimicrobial peptides called defensins and phagocytosis and the complement system. This type of protective mechanism is present by birth and is known as innate immunity. Jarred vertebrates, including humans, have complex defense mechanisms. The typical vertebrate immune system consists of many types of proteins, cells, organs, and tissues that interact in a complicated and vigorous network. As part of this more complex network response, the human immune system adapted over time to recognize specific pathogens more competently. This adaptation process is referred to as adaptive immunity or acquired immunity and generates immunological memory. Immunological memory generated from a primary response to a specific pathogen provides an enriched response to secondary encounters with that same pathogen. This process of acquired immunity is the basis of vaccination. Primary response can take 2 days to 2 weeks to develop. After the body gains immunity towards a certain pathogen, when infection by that pathogen occurs again, the immune response is called the secondary response. So you could see a flowchart showing the components of immune system. Immune system: innate, non-specific, first line of defenses; then adaptive, specific, second line of defenses, protects or re-exposes. First, innate immune system, cellular components are there and humoral components are there. But in case of adaptive immunity, cellular components are also there. So similarly, there must be some sort of immune components. First is about innate immunity. Microorganisms are pervasive. They are in the air we breathe and in our on the food we eat. This our epithelial surfaces like skin, respiratory tract, gastrointestinal tract, genitourinary tract are continuously exposed to microorganisms. Diseases occur when microorganisms are able to invade epithelial surfaces. In view of our constant exposure to microorganisms, it is surprising that normal healthy person experiences long infection-free periods, and these infections are the exception rather than the rule. This persistent or the resistance or immunity of epithelial surfaces to invasion is a characteristic which is present from birth and is therefore called innate or natural immunity. The elements of the innate or non-specific immune system include anatomical barriers, secretory molecules, and cellular components. Among the mechanical and anatomical barriers are the skin and internal epithelial layers, the movement of the intestines, and the oscillation of bronchopulmonary cilia. Associated with these protective surfaces are chemical and biological agents. The elements of the innate non-specific immune system include anatomical barriers, secretory molecules, and cellular components. Among the mechanical and anatomical barriers are the skin and internal epithelial layers, the movement of the intestine, and the oscillation of bronchopulmonary cilia. Associated with these protective surfaces are chemical and biological agents. Anatomical barriers to infections. Mechanical. The epithelial surfaces form a physical barrier which are impermeable to most infectious agents. The skin acts as our first line of defense against any invading organisms. The desquamation of skin epithelium also helps in removing bacteria and other infectious agents that have attached to the epithelial surfaces. Movement of cilia or peristalsis helps in keeping air passage and the gastrointestinal tract free from microorganisms. The flushing action of tears and saliva helps in preventing infections of the eyes and oral cavity or mouth. The mucous lining of the respiratory and gastrointestinal tract traps the microorganisms and helps in protecting the lungs and digestive systems from infection. Next, chemical. Fatty acids in the sweat inhibit the growth of bacteria. Lysozyme and phospholipase found in tears, saliva, and nasal secretions can break down the cell wall of bacteria. The acidic pH of sweat and gastric secretions prevent growth of bacteria and many other microorganisms. Deficits, low molecular weight proteins found in the lung and gastrointestinal tract are known to have antimicrobial activity. Surfactants in the lung act as opsonins, molecules that promote phagocytosis. Opsonins. Surfactants in the lung act as opsonins. That is a new information for me. Deficits are there, which are low molecular weight proteins found in the lung and gastrointestinal tract, are known to have antimicrobial activity. Then same is the case with surfactants, but they act as opsonins. Opsonins are molecules that promote phagocytosis. Deficits and surfactants. The normal, next we will be learning biological aspects. So the normal microbial flora of the skin and the gastrointestinal tract prevents the establishment of pathogenic bacteria by secreting toxic substances or by competing with pathogenic bacteria for nutrients or attachment to cell surfaces. Then humoral barriers to infection. The anatomical barriers are very effective in preventing colonization of tissues by microorganisms. However, when there is damage to tissues, the anatomical barriers are infringed and infection may occur. Okay. Once infectious agents are breached tissues, another innate defense mechanism comes into play, and that is the acute inflammation. These humoral factors are found in serum. Complement system. The complement system is a major humoral, non-specific defense mechanism. It is responsible for the increased vascular permeability, recruitment to phagocytic cells, and lysis and opsonization of bacteria or microorganisms. Complement system. The complement system is a major humoral non-specific defense mechanism. Non-specific defense mechanism, that is a humoral one. It is responsible for the increased vascular permeability, recruitment of phagocytic cells, and lysis and opsonization of bacteria microorganisms. Coagulation system. Depending on the gravity of the tissue injury, the coagulation system may or may not be activated. Some products of the coagulation system can contribute to the non-specific defenses because of their ability to increase vascular permeability and act as chemotactic agents for phagocytic cells. In addition, some of the products of the coagulation system are directly antimicrobial. Then lactoferrin and transferrin. These proteins, by binding with anion, which is an essential nutrient for bacteria, downregulates the growth of bacteria. Lactoferrin and transferrin. Then interferons. Interferons are proteins that can control virus replication in cells. Then lysozyme. Lysozymes break down the cell wall of bacteria. Then interleukin 1, IL-1. IL-1 induces the production of acute phase protein, which are antimicrobial as they can opsonize bacteria. Then cellular barriers to infection. Part of the inflammatory response is the employment of polymorphonuclear eosinophils, neutrophils, and macrophiles to sites of infection. These cells are the main line of defenses in the non-specific immune system. Cellular barriers to infection. The neutrophils. These cells are engaged to the site of infection where they phagocytose invading microorganisms and kill them. In addition, they also contribute to collateral tissue damage that occurs during inflammation. Then macrophages. Tissue macrophages function in phagocytosis and intracellular killing of microorganisms. Furthermore, macrophages contribute to tissue repair and act as antigen presenting cells or APCs, which are required for the introduction of specific immune response. And natural killer cells and lymphokine activated killer cells or LAC cells. NK and LAC cells can non-specifically kill virus infected and tumor cells. These cells are important in non-specific immunity to viral infections and tumor surveillance. Then eosinophils. The granules of eosinophils have lytic properties that help in killing certain parasites. Phagocytic barriers. Phagocytosis from Greek word phago means eating, cyte means vessel, and osis means process. Is the cellular process of engulfing solid particles by the cell to form an internal phagosome. Phagocytosis is a specific form of endocytosis involving the vesicular internalization of solid particles such as bacteria. So, phagocytosis in the immune system is a major mechanism used to remove pathogens and cell debris. Bacteria, dead tissue cells, and small mineral particles are all examples of objects that may be phagocytosed. It's a diagram which depicts the process of phagocytosis. If we kind of zoom this, we would be able to read. Next is about inflammatory barriers. Yeah, I was trying to read this. Yeah. Waste materials discharged. Yeah, it's reverse process. Bacteria attachment. Phagocytosis. Phagocytosis produced lysosome with digestive enzymes. Golgi apparatus produce these lysosomes. Nucleus phagocyte digested molecule. Endoplasmic reticulum is also there. Waste materials discharged into the outside. Outside in the sense into the cytoplasm. Yeah, extracellular matrix, I guess. And inflammatory barriers. When the body is injured, a sequence of events is started that ultimately leads to the repair of the site. The inflammatory response is the body's natural response that occurs immediately following tissue damage. Its main functions are to defend the body against harmful substances, dispose of dead tissue, and to promote the renewal of normal tissue. The inflammatory reaction is characterized by four distinct signs, each of which is due to a physiological response to tissue injury. Due to chemicals released by damaged cells, tumor, swelling, or edema due to an influx of fluid into the damaged area, then redness due to rubor or redness due to vasodilation, the widening of tissue blood vessels, and calor or heat due to an increase in blood flow to the area. The inflammatory reaction is the combination of a number of overlapping reactions within the body. Although a lot of these occur simultaneously, but a certain order of events are there. So, like tissue injury, tissue damage may occur from number of reasons, like release of chemicals. And the cells of a tissue are injured. There is a release of number of chemicals that initiate the inflammatory response. Examples of these are kinins, prostaglandins, and histamine. These chemicals work collectively and are responsible for increased vasodilation or widening of blood capillaries and permeability of the capillaries. This leads to increased blood flow to the injured site. These chemicals work collectively and are responsible for increased vasodilation or widening of blood capillaries and permeability of the capillaries. This leads to increased blood flow to the injured site. These substances also act as chemical messengers that attract the body's natural defense cells. This process is known as chemotaxis. Some chemicals also increase the sensitivity of the pain fibers in the area, and so the area becomes painful. Then leukocyte migration will be there. Chemotaxis leads to the migration of certain white blood cells or leukocytes to the damaged area. Two types of leukocytes that are predominantly involved in the immune response are macrophages and neutrophils. Neutrophils are first to migrate to the injured site and function by neutralizing harmful bacteria. Macrophages aid in the healing process. Neutrophils are first to migrate to the injured site and function by neutralizing harmful bacteria. Macrophages aid the healing process by engulfing bacteria and dead cells and ingesting them so that the area is clear for new cells to grow. They arrive at the injured site within 72 hours of injury and may remain in the area for a week after the injury. There's a pictorial depiction of the process of inflammation. Bacteria and other pathogens in the wound region. Platelets form blood release blood clotting proteins at wound site. Mast cells secrete factors that mediate vasodilation and vascular constriction. Delivery of blood, plasma, and cells to injured area increase. Neutrophils secrete factors that kill and degrade pathogens. Neutrophils and macrophages remove pathogens by phagocytosis. Macrophages secrete hormones called cytokines that attract immune system cells to the site and activate cells involved in tissue repair. Inflammatory response continues until the foreign materials is eliminated and the wound is repaired. Blood platelets, mast cells, neutrophils, cytokine signaling path to injury site, cytokines innate tissue repair, macrophages. Once again, bacteria and other pathogens enter a wound. Platelets from blood release blood clotting proteins at wound site. Mast cells secrete factors that mediate vasodilation and vascular constriction. Delivery of blood, plasma, and cells to injured area increase. Neutrophils secrete factors that kill and degrade pathogens. Neutrophils and macrophages remove pathogens by phagocytosis. Macrophages secrete hormones called cytokines that attract immune system cells to the site and activate cells involved in tissue repair. Inflammatory response continues until the foreign material is eliminated and the wound is repaired. The adaptive immunity or specific immunity is comprised of highly specialized immune cells and processes that remove or check the pathogenic growth. This type of immunity works to protect and heal the body when the innate immunity fails. It provides the body with the ability to recognize and remember pathogens and other antigens. This mechanism allows the immune system to mount stronger attacks each time the pathogen is encountered, thus repairing itself for future challenges and preventing reinfection by the same pathogen. The four hallmarks of the adaptive immunity are immunological recognition, self non-self discrimination, immunological specificity, immunological memory, and immune response must recognize a microorganism as foreign or non-self as distinct from self, respond to a microorganism by production of specific antibodies and specific lymphocytes, mediate the elimination of microorganism. An agent which evokes an immune response is called an immunogen. An agent which evokes an immune response is called an immunogen. The term antigen is applied to a substance which reacts with antibody. An agent which evokes an immune response is called an immunogen, and the term antigen is applied to a substance which reacts with antibody. Innate immunity, physical barriers, cellular, chemical barriers. Physical barriers in the sense skin mucous membranes, cellular, PMNs, monocyte, macrophages, and eosinophils. Chemical barriers in the sense pH, lipids, enzymes, etc. So PMN, monocytes, macrophages, eosinophils, acquired immunity, humoral B cells, antibodies, cellular T cells, lymphokines. Humoral B cells, antibodies. Just remember a human body. Body in the sense B for B cells, humoral H for human. Like that. It's a way to remember that. Another, the opposite, cellular T cells, lymphokines. Immunity, active, passive, and delayed. Active, passive, and delayed immunity are all variations of preparation of immune system. Thereby, antibodies are produced in response to the presence of an antigen considered to be foreign. Active and passive immunity are versions of what is known as antibody-mediated immunity. That is, antibodies bind to the antigen and this binding further stimulates the immune response to respond to the antigen threat. Antibody-mediated immunity is also called a humoral immunity. Active immunity. Active immunity ensures due to the production of antibodies. Antibodies are produced as a result of the presence of the target antigen either as part of an infecting organism or because of the introduction of a specific antigen. The immunity is provided by an individual's own immune system. The type of immunity invoked by the active response tends to be permanent. The immune system has a capability or capacity for memory of the antigen. If presented with the same antigen challenge again, the immune system responsible for the formation of the corresponding antibody is rapidly elicited. An example of active immunity is the injection into healthy individuals of the disabled toxins of bacteria such as Conibacterium diphtheria, the agent causing diphtheria, and Clostridium tetani, the bacteria that causes tetanus. Passive immunity is also because of the presence of antibody, but the particular individual does not produce the antibody. Rather, the antibody is produced in someone else and is introduced into the recipient. An example is the transfer of antibodies from a mother to the unborn child in the womb. Such antibodies confer some immune protection to the neonate in the first 6 months before birth, following birth. Indeed, the transient nature of the protection is an hallmark of passive immunity. Protection fades over the course of weeks or a few months following the introduction of a particular antibody. For example, a newborn carries protective maternal antibodies to several diseases, including measles, mumps, and rubella. But by the end of individual's first year of life, vaccination with the NMR vaccine is necessary to maintain the protection, or it's kind of short lived. Another example of passive immunization is the administration of humans of tetanus antitoxin that is produced in a horse in response to the inactivated tetanus toxin. This procedure is typically done if someone has been exposed to a situation where the possibility of contracting tetanus exists. Rather than rely on individual's immune system to respond to the presence of toxin, neutralizing antibodies are administered right away. Then delayed immunity. A third type of immunity, which is known as delayed immunity, is a different sort of immunity. Delayed immunity is a so-called cell-mediated immunity. Here, immune responses called B cells, immune components called T cells, bind to the surface of other cells that contain the antigen on their surface. This binding triggers a further response by the immune system to the foreign antigen. The response can involve components such as leukocytes. An example of delayed immunity is the tuberculin test or the Mandox test, which tests for the presence of Mycobacterium tuberculosis, the bacterium that causes tuberculosis. A small amount of bacterial protein is injected into the skin. If the individual is infected with a bacteria, it has ever been infected, the injection site become inflamed within 24 hours. The response is delayed in time relative to the immediate response of antibody based immunity, hence the name of immunity. Active immunity, naturally acquired, artificially acquired. Passive immunity, naturally acquired, artificially acquired with the help of serum. So now we are gonna have a quickly, I'm sorry, a quick summary of all this topic we mentioned earlier. So let's try it. History of immunology dates back to the series which were he wrote in 430 BC that only those who had survived the plague can nurse the sick because they would not contract the disease a second time. Chinese and Turks in 15th century started the process of variolation in order to protect themselves from diseases. Lady Mary Wortley Montagu, I'm not sure about the pronunciation, variolated her own children, and the technique was popularized in Great Britain. Vaccination, the word Jenner invented for his treatment, was adopted by Louis Pasteur for immunization against any disease. Louis Pasteur developed his germ theory of disease. Later on, Metchnikoff described the roles of cells, and at the same time, Bering described the role of serum components in providing the immunity. Out of various theories for immune response, clonal selection theory of Burnett is widely accepted theory of generation of immune response. Invertebrates has two types of immunity: innate, non-specific, and adaptive or specific. Innate immune response begin much more quickly than adaptive ones and therefore constitute the first line of defense after the host has been invaded by a pathogen. Innate immune response include various barriers such as anatomical, physiological, chemical, phagocytic, and inflammatory. Adaptive immune response exhibit four antibody attributes: specificity, diversity, memory, and recognition of self and non-self. Active, passive, and delayed immunity are all variations on the operation of the immune system, whereby antibodies are produced in response to the presence of an antigen considered to be foreign. Active immunity occurs due to the production of an antibody as a result of the presence of target antigen, either as part of an intact infecting organism or because of the production of the specific antigen in the form of a vaccine. The immunity is provided by an individual's own immune system. Passive immunity is also because of the presence of antibody. However, a particular individual does not produce the antibody, rather, the antibody which has been produced in someone else is introduced into the recipient. So that's all about this lecture, and I once again want to thank the creators of this PDF. Thank you. Thank you so much. Thank you for listening.